So yeah, hey everybody, happy week 3. I hope everyone's doing well. So thanks everyone for seeing my many emails and sorry for bombarding you. But yeah, this week we're doing our special topics lecture at a different day and time. Next week we'll revert back to the original time. So it's my pleasure to introduce Dr. Alex Chavez, MD, PhD. He's an associate professor in the Department of Pediatrics at UCSD. So his lab, he's a faculty member in the bioinformatics program, and his lab is interested in building technologies that solve critical research bottlenecks and answer fundamental biological questions. And kind of as you've been seeing over the past couple of weeks, each of our special topics lectures does something in the intersection of computing and biology. A lot of them do biomedical applications. Some of them are more on the computational side, some of them more on the biological side, some more in medicine. So yeah, hopefully, everyone's excited to hear Dr. Chavez talk about his cool research. Yeah, thanks for being with us.
Cool. Yeah, thanks for the intro, Nima. Yeah, and honestly, if people want to interrupt along the way, that's great. If not, I talk really fast, so we'll definitely be on time if no one interrupts me. So with that, I'll just kind of dive in. Yeah, so I'm Alex, and thanks for giving me the opportunity to share our research with you. And so going forward, just a few of my conflicts of interest, and kind of keep moving. So overview for the talk, tell you about some of our work on multiplex drug screening, tell you about our work characterizing SARS-CoV-2 3CL protease, and finally, our work on examining how SARS-CoV-2 adapts to drug. And with that, we'll start on this multiplex drug screening work. I always like to point out that it was done by a very talented graduate student in the lab, Debbie Hong, along with a postdoc and a research technician, Jiwan Cha and Chris Fazakis. So a little bit of background on drug screening for the group. So typically when you're screening for new small molecules against a protein of interest, you start with that protein and you screen hundreds of thousands of compounds against it. And this has been quite effective, but it takes quite a bit of time, money, and effort to get the answer to one thing, i.e., what is the compound that inhibits this one target? And while that's been very effective, it's a little bit of a problem for viruses because there's hundreds of viruses that currently infect humans, and there's tens of thousands of viruses that circulate in animals that have the potential to jump into humans. And so while it's been amazing to find drugs against things like SARS-CoV-2, ideally what we want to find is maybe more broad-acting inhibitors that work against multiple different coronaviruses in case, as what happened recently, we have another pandemic. And so what viral proteins are our lab interested in looking for inhibitors against? We're focusing on viral proteases, and we're doing that because 50% of viruses encode a protease. They're required for viral replication, so the protease is dead, virus doesn't live. And there's been a lot of success of protease inhibitors in HIV and hepatitis C treatment, and in addition, now also against SARS-CoV-2. And so our goal when we started this project about 5 years ago was to identify broad-acting protease inhibitors. And so to achieve that goal, we decided that we needed to change the way that drug screens were performed. So we needed to develop some sort of method that let us look at not just one viral protease at a time, but hopefully dozens. And so how is it that we screen for protease inhibitors? We use a very simple system, and that's the mighty little yeast. And so it turns out if you take a little yeast cell and you make it express a viral protease, it hates it. It basically doesn't grow and it's very sad, very sick. You can then take that little sad, sick yeast cell expressing a viral protease and grow it in the presence of a protease inhibitor, and now it becomes happy again and grows really well. And so this lets you use yeast almost as a biosensor of protease activity. Protease active, yeast sad. Protease inhibited, yeast happy again. And so now we can make lots of these little biosensor yeast by just taking different viral proteases and sticking them inside of different yeast cells. So that's great. So now we have a lot of ways to basically build these little sensors for protease activity, but how do we study them all at the same time? And so to do that, we use DNA barcoding technology. So here's 4 little yeast cells, and here's 4 little DNA barcodes. And what we do is we take these DNA barcodes and we insert them into the genome of yeast into a precise location. And for each yeast cell, we know exactly what barcode they're getting and where it's going. And so going forward for the remainder of this talk, when you see a different colored yeast cell, represents a cell that has a barcode that we know its identity. And so now what we can do is we can take the little blue yeast and we can make them express SARS-CoV-2 3CL protease. We can take the teal yeast, make them express MERS 3CL protease. Take the black yeast, make them express Zika protease. And then as a control, we'll take the yellow yeast and make them express a non-toxic gene called EYFP. This gene actually is a fluorescent protein. It basically makes the yeast yellow and they don't mind being yellow. And so now we have this little pool of yeast cells, and we can now make them all express these proteins that we give them. And what we see is that all the yeast cells expressing the proteases are sick, and so they deplete in the pool. All the yeast cells that are yellow don't mind it, and they grow really, really well. And so to see that change in the distribution of the library, we basically explode all these cells open, and we sequence out those little barcodes that were inside of them. And then this is where basically we're counting how many times each barcode exists in that pool. We can now go back to our initial pool of cells, induce the protease expression, this time out of small molecule. And if we get a result like this, it suggests that now that the SARS-CoV-2 and the MERS 3-cell protease expressing cells are growing better, that compound that we added might be inhibitor against that. And so this here is sort of where the computation for our work occurs, where basically we're counting the barcode conditions in the drug situation and comparing that to the barcode conditions without the drug. And we're saying, is there a difference between those? And one other thing that we're doing that I don't mention is that every model is not given just a single barcode. It's put into 5 independent barcoded cells. So we look for the consensus for all 5 barcodes associated with one protease and ask, are they all going the same direction? What's that magnitude? And that's all the kind of computational pipeline on the back end that makes this all really work. Great. So does this idea actually function? So I'll show you a little focused study. In this case, I'm showing you a pool of 30 proteases tested against 96 compounds. That's 2,880 tests performed in a single 96-well plate. And I'll only show you a subset of the data, so I'll zoom into that. On the x-axis are different viral proteases that are inside of yeast cells. On the y-axis are different compounds we're testing those yeast against. And you'll see these different colored squares represent when there's a hit. So if there's a black square, it means that that compound rescued the growth. And basically, we see everything that we expect. MAC 5576 and GC376 are known coronavirus protease inhibitors, and they're rescuing the growth of those yeast cells expressing the coronavirus proteases. Lopinavir and darunavir are known HIV protease inhibitors, and they're rescuing the growth of the yeast cells expressing HIV protease. Glecaprevir is a known hepatitis C protease inhibitor, and it's rescuing the growth of the hepatitis C expressing protease expressing yeast cells. In addition, when we test other non-protease inhibitory compounds like PARP inhibitors and kinase inhibitors, we see that they do not rescue any of the models, showing us that it's not just random signal we're getting. The signal seems to be catered towards a true inhibitor being detected. So what does our current assay look like? We currently have a pool of 40 viral proteases that span various viral families. And we also have 4 controls within our pool that help us basically just normalize all our data. And we've been screening that against a library of diverse compounds. And here's some of our initial data. On the x-axis are a bunch of different protease models in our pool. On the y-axis are a bunch of compounds that we tested. And we got this data, we saw this block over here that we thought was quite interesting, where there was a series of compounds that seemed to inhibit a lot of different 3CL proteases from coronaviruses. And we looked at the structure of those compounds. We noticed that they looked quite similar. They had this little like 2-member ring over here. And we thought, oh, these are kind of all structurally similar. Maybe make sense that they're behaving a little bit the same. And so that's great. The screen, this kind of complex multiplex DNA barcoding thing said that things were working, but how do we really know that it was working? So to test that, we took those individual compounds and we took a single yeast cell expressing SARS-CoV-2 3-cell protease, and we tested different dosages of the drug against that model. So now no fancy barcoding, just one at a time testing. We grow the yeast cells in different concentration of drug, and we look to see, do we see a response where as we add more drug, the yeast get happier? Because that's what we'd expect. This is a true inhibitor. The compound's blocking the protease. As we add more compound, the yeast should get happier, happier, and happier. And that's literally what you see, where you see this kind of curve increases and slopes upward as we add more and more compound. And so, and that has a rescue of like 1 micromolar. It's a half concentration rescue. And if you look at 2 other, the compounds that look kind of similar to it, again, when we do these little dose response curves, you see a similar sort of rescue. And they're all kind of rescuing at like half a low micromolar range. So that makes us feel that, our crazy multiplex screen actually worked, and that when you test things one at a time, you get the same sort of results. Again, the screen said that these compounds inhibited many, many 3-cell proteases. And so we actually manually went ahead and tested all of those against these compounds. And again, we saw that basically manual testing recapitulated this multiplex screening approach. So that's very nice. And that's kind of nice to see that we can rescue this kind of weird model where we express viral proteases inside of yeast. But how do we know that's actually real? How do we know that's not rescuing something else? Maybe it's just making these cells not mind having the protease inside them. Maybe it's just like making these cells not die because that's there. And so to test that, we actually purified the SARS-CoV-2 3-cell protease. And then we tested that purified protease, no cells, no nothing, just that purified protein against these same compounds. And when we did that, again, we saw beautiful dose response curves, where as we add more of that compound, we see more inhibition of that purified protease. The same thing for the other 2 members that look similar to it. And so this kind of told us that this represents sort of a bona fide inhibitor, that this compound really is directly acting on the protease to mediate the effect that we see. And so diving a little bit into the mechanism, our chemical collaborators looked at the compound, they thought it might be a covalent inhibitor, and they thought that this is what actually happened, that this compound basically kind of attacked a critical cysteine within the protease, and it formed a sulfur bond. And so to test that, you can literally purify the protease, and you can run it on LC mass spec, and you can basically like quantify its weight. And so this is, in the no compound condition I'm showing you, this is the exact weight of the protease when you like purify it from cells. If this chemical compound forms a covalent bond to the protease, it should shift its weight by about 95 Daltons, and that's exactly what we see. So now when we incubate the protease with our compound, we see that the protease shifts its weight by about the weight we expect if this compound were to become covalently attached to it. And we've also been working with some other collaborators to actually crystallize this compound acting to 3CL protease. And there's some initial data suggesting how it might be binding into the pocket of the protease and inhibits its activity. So next steps for this project, we want to screen additional compounds using our method. We want to further develop some of our inhibitors that we found to make them more potent. And we want to apply this platform to other essential viral proteases. And so summary for part one of my talk, described how we can use DNA barcoding to study dozens of proteases at once. Despite the scale that we're operating at, where we're testing 40 proteases at the same time, we get real hits. And we validated a set of broad-acting inhibitors against the 3CL proteases from coronaviruses. So before I move on, I always like to pause at this point and ask if there are any questions.
Any questions, folks? Feel free to type in the chat. Feel free to unmute. Questions?
Or clarifications also totally fine.
Looks like we're good.
Cool. I'll keep going then. Great. So I'll move on to the next part of the talk, characterizing the SARS-CoV-2 3CL protease. And so this is work that's done by 3 really talented PhD students in the lab, Sho, Debbie, and Jenny. And so background on COVID-19. According to CDC, COVID-19 has caused over 6.3 million hospitalizations and 1.1 million deaths in the US. Vaccines obviously transformed the disease course, and they've prevented an estimated 3.2 million deaths. But not everyone's vaccinated. Sadly, 5% of Americans over 65 have not received a single dose of vaccine. In addition, not all patients that are vaccinated actually respond to it. For example, people that are immunosuppressed, such as those on chemotherapy or with other disorders that inhibit their immune system. In addition, as we all know, the virus continues to evolve and has managed to escape immunity, and this has decreased vaccine effectiveness. And so for all these reasons, we need alternative ways to fight the virus. So what other options exist to fight the virus? There are now several FDA-approved chemical inhibitors against SARS-CoV-2. One of them that I would argue is probably the most popular is Paxlovid, which is a orally bioavailable, highly effective protease inhibitor. It's caused in the initial clinical trial, reduced the rate of severe COVID by about 90%. And the way that Paxlovid works is that it inhibits viral 3CL protease. And so that protease is required for the virus to create functional proteins. And so what it literally does is the virus actually makes one super, super long protein that the protease comes in and chops into little pieces. And if that protease is inhibited, the virus just has one crazy, crazy long polyprotein that's just non-functional. And so this compound basically blocks the protease, and by doing that, it makes the virus not be able to make anything functional. And so what are the motivations for our study? When we started this work, we knew that the viral spike protein, which is the target of the antibodies in the vaccine, had this incredible ability to mutate. And that has made vaccines less effective. So we asked, does the 3CL protease, this target of Paxlovid, have the same ability to mutate? And if it does, could we use our understanding of the virus's tolerance to mutations to predict how it might become resistant to drug? And so question one is 3CL protease able to tolerate being mutated? And so to test that, we did what's called deep mutational scan. We mutated each amino acid of the protease to all other possibilities. And so the protease is 306 amino acids large. And so we made 6,120 unique variants of the protease and tested them all at the same time. And then we asked which variant remains active. And so again, this is really the part of the project that involved the computation where we got all this data back for these thousands of different variants. And we need to understand which were more or less functional than the wild type version, and also try to understand how they might be working to cause this effect. So here's a bird's eye view of the data. On the x-axis are all the different amino acids in the protease. On the y-axis are all the possible variants that could exist at that location in the protease. You'll see different colored squares. The blue squares mean that this protease variant is more active than the regular version of the protease. The red squares mean that this variant is less active than the regular protease, and the white squares mean that these mutations are neutral, has no effect on the protease, works just like wild type version. So these are what all the data look like, and it's a lot, so I'll kind of walk you through it. Basically, this top row here represents what happens if I insert a stop codon into the protease. Well, you might imagine if you insert a stop codon, well, you're not making the full protease then, so like it's not going to work. Except what's kind of cute is you can kind of chop off the little end of the protease, and it doesn't seem to matter. So if you do that, you notice that all those little squares are kind of like this white kind of neutral color. In addition, you see these two big chunks in the protease where you can mutate them quite a bit, and the protease doesn't seem to mind. It kind of just like hangs out, sort of chilling. But there's this kind of region in the middle where if you mutate it, the protease really doesn't like it, and there's sort of like kills it. And that's kind of this block of like darker color in the center. Great, so we got that data back. We're like, okay, that's really pretty, but like, what does that actually mean? And so to understand that, we decided to map our activity onto the structure of the protease. So this is actually what the SARS-CoV-2 3-cell protease looks like. It's a homodimer, so that means that it's the same protein kind of repeated and bound to itself. And so onto this structure, we basically colored things by if they tolerate mutations or don't tolerate mutations. So regions of the protease that tolerate mutations are colored in blue. Regions of protease that hate mutations are colored in red. And so right here is the catalytic pocket of the protease. This is the part of the protease that actually chews on other proteins. And so this is the deep mutational scan that we produced, the data I kind of showed you previously. And now let's map that data onto the structure together. And so you see this region over here, this kind of like the first pocket where it didn't mind being mutated. You see when you map it on the structure, it's quite far away from the catalytic pocket, sort of on the outside edge of the protease. This other pocket that didn't mind being mutated, again, also on the outside edge, far away from the catalytic pocket. And this region right in the center that hated being mutated is kind of right there in the catalytic pocket, or in what's called the dimerization interface, the region of the protein where each of the pieces come together to interact. And so our map looked kind of reasonable, but how do we know it's actually telling us truth? And so to test that, we basically plotted all the different protease variants we had studied, and I'm showing you how many on the y-axis. I'm plotting that, and the x-axis I'm plotting you their activity score. And so when we plot our data onto this chart, we see something that looks like this. And so basically what we see is there's a set of protease variants that have very low activity, and we consider them to be inactive. And a set of protease variants that have very high activity and consider them to be active. And so I guess sadly, we've actually had to sequence millions of variants of the SARS-CoV-2 that we've isolated from patients. And from all that sequencing data, there are people out there that just have like random mutations. So the virus is just kind of like randomly mutating all the time. And sometimes it randomly generates a mutation in the protease. If that mutation is functional, the protease keeps working and it can cause illness in someone. But if it has a mutation that's non-functional, then that protease should not exist and we shouldn't see it in people. So basically, everything that we see in people, we think represents a version of the protease that is totally active and that works. And so we asked, how do all the existing variants of the protease that we see in people, how are they scored in our assay? Where do they belong on this graph? And basically, our assay scores the vast majority of variants that we see inside of humans as active. So that argues that our assay is actually seeing true activity of the protease and not some quirky aspect of the screen that we did. And so now that we have this database of how the virus can tolerate mutations, can we use it to predict drug-resistant variants? So to do that, we looked at the crystal structure of SARS-CoV-2 3-cell protease bound to the active ingredient Paxlovid, which is called nirmatrelvir. That's actually the compound that is the protease inhibitor within that cocktail. And we identified there are a series of key residues that actually mediated that interaction between the drug and the protease. We then asked, can any of these residues that mediate these key interactions be mutated according to our screen? And what we found is that this glutamate 166 actually in our screen was highly tolerant to mutations, i.e. you could mutate it in the protease and didn't seem to care, but it would be predicted that if you mutated it, it would reduce its ability for the compound to bind to the protease. And so to test that, we basically made a recombinant version of the virus where we mutated residue E166 from glutamate to valine, so very, very different amino acid. And then we asked, does that now make the virus resistant to drug? So on the x-axis, I'm showing you increasing concentrations of the drug, of the active ingredient in Paxlovid. On the y-axis, I'm showing you the inhibition of the virus. So if the virus is 100% inhibited, you'll be on the top of this graph. And so when you take the wild-type virus and add increasing concentrations of Paxlovid, you basically see that you inhibit the virus more and more and more. And that at about 53 nanomolar, you can inhibit about half the viruses growing inside of the cells. In contrast, when you make the E166V mutation, you see a marked decrease in its response to drug, and it takes about 200 times more drug to inhibit the virus. And so this really suggests that this mutation that we predicted actually does confer drug resistance, and we were able to use our mutational scan to discover it. So next steps for this project, we want to use this mutational map to guide future drug development efforts. And to do that, we want to actually have drug developers try to avoid having drugs bind to the residues that we identified as flexible. We now know which residues in the protease can easily mutate away, and there are also other residues that it can't easily mutate away. And so if you had a compound that bound to the residues that basically the protease can never mutate away, then that compound in theory should be more resistant to actually having resistance develop against it. We also want to take this approach of deep mutational scans and apply to other viral proteins to understand their function and also potentially explore their druggability. Because again, if you have a viral protein that you can basically mutate like crazy and it's still functional, that might be a poor drug target, because once you have a drug try to bind to it, it'll quickly just mutate away to avoid that drug. So summary for part 2 of the talk, we created this comprehensive mutational map of SARS-CoV-2 3-cell protease. We showed that our screening results correlated very well with clinical data from real people, and we used that data to predict drug resistance conferring mutations. And so again, I'll pause at this intermediate point and see if there are any questions.
Questions from folks? It's a once-in-a-lifetime opportunity. We got an expert here. Feel free to type in the chat. Feel free to unmute.
It's all good. It's all good. We're feeling shy today. I like it.
No worries. Yeah, maybe at the end of the talk, there will be more questions.
Sounds good. Cool. All right. And then for the last part of the talk, I'll talk about our work examining how the real virus adapts to drug. And this is work that was carried out again by 3 really talented graduate students, Sho, Debbie, and Bruce, along with a collaborator, Dr. David Ho at Columbia University. And so our previous data had shown that E1C6B caused resistance to the drug Paxlovid, but is this the only way that the virus can get resistance to that compound? Our mutational scan suggests that there's lots of flexible residues in the protease, and so we were wondering, how does the virus take advantage of this natural flexibility to gain resistance? Are there many routes to resistance, or are there only one or a very few number? And so to study the way that the virus adapts to drug, we basically took a strategy of growing the virus and increasing concentrations of the drug. And so we started with the wild-type population of the virus, threw it onto cells with a low dose of the active ingredient Paxlovid, and then we took the viruses that grew out of that, threw them onto cells again, this time with increasing more concentration of drug, and kind of repeated that process over and over again, each time adding more and more drug. To the point at the end of this, we get virus that's highly resistant to nirmatrelvir. And so usually when people do these sort of studies, they'll take 2 or 3 isolates of virus and kind of carry it through this passaging scheme. But we really wanted to have a more global view to how the virus sort of evolves resistance. So to do that, we took 53 isolates of the virus and put them through this whole scheme. And in addition, we sequenced the virus at 4 different time points. And it produced this chart where we basically are able to catalog the mutations that occurred during the virus's growth with drug. And so again, this is the point of computation in our lab, where we got all this DNA sequencing back from all these different viral genomes. We needed to figure out which were the residues that were enriching and what percentage were they enriching within the population. And so what we found when we looked at our data was that normally the virus obviously starts in the wild type situation, and then there's a series of mutations that occur very early on, with about 3 mutations being very, very commonly the ones that we see occurring initially. Over time, as we continued to grow the virus and increasing concentrations of drug, we saw other mutations would emerge. And when we looked at this data as a whole, what we saw was that some mutations would arise early. For example, this P252L mutation always arose early in our passaging scheme, while other mutations arose later, like this L50F. And so what this told us is that there are some mutations that on their own are likely deleterious, and they need other mutations to be there first in order to make them viable. So essentially, there are mutations that are called gateway mutations. They can sort of open up doors to let other mutations then become feasible. And so L50F is one of these mutations that needs a gateway mutation to open up a door to allow it to occur. In addition, when you look at all the mutations that we found, the virus doesn't seem to have a lot of mutations that it likes to use. In fact, there's a kind of a small number of cards that it just shuffles in different ways to confer resistance. So it's not like there's hundreds of different mutations that we saw enriching. There's kind of a much smaller subset. And so again, how do we really know that these data that we generated are actually real? We were passaging this virus over the course of 2 months and increased concentration of drug. maybe there's other crazy mutations that occurred in that virus that were outside of the protease that we were just ignoring, or there's some other weird adaptation that the virus had done to cause resistance to drug outside of mutating the protease. So to test that, we basically made fresh recombinant virus, where we take wild-type virus and use genetic engineering to insert a single mutation or a couple mutations into the virus. Everything else, though, is the original sequence. And then we can test those recombinant viruses for the resistance to drug. Showing you an example here, if the virus has a T21I mutation, we find that when that mutation is there by itself, confers about a 5-fold resistance to drug. Here's what the rest of the single mutations look like. Again, most of them incur about a 2 to 5-fold increased drug resistance. But we start to get the pairs of mutations, you start to see this sort of synergistic additive effect on drug resistance, which is exactly what we saw, that over time, the virus accumulated more mutations and was able to grow in higher and higher concentrations of drug. And so that was interesting, but one question that we wanted to ask ourselves is, okay, we've now got these viruses that are resistant to this drug nirmatrelvir. Are they just generally resistant now to all protease inhibitors, or are they very specific in what they're resistant against? So there's another drug called ensitrelvir that's currently in phase 3 clinical trials that is also a SARS-CoV-2 3CL protease inhibitor. And it turns out when you look at the mutations that we found that confer resistance to nirmatrelvir, some of them do confer resistance to ensitrelvir, but others don't. So this really suggests that we've identified real mutations that cause resistance, and there's no universal set of mutations that confer resistance against all drugs, although there are some that confer sort of like partial resistance. And so next steps for this project, we've been actually monitoring SARS-CoV-2 clinical sequencing to see if any mutations that we found in our assay are starting to enrich in the general population, i.e. when you look at secret circulating viruses now, are they starting to get these like E166V mutations that make the virus very resistant to Paxlovid as more and more people are taking this drug? And luckily, what we found is that none of these mutations that we've identified have actually enriched strongly in the general population. Although there have been case reports, these are basically clinical reports from hospitals where they've been following patients that have been taking Paxlovid for like 2 months, 3 months. And when they sequence the virus inside those people, they find the very same mutations that we found in the laboratory occurring in those patients, arguing that now someone's sort of done the human experiments, and that what we found in the lab is being recapitulated within people. So summary part 3 of the talk, we passed through the virus at multiple increasing concentrations of drug. We used that to identify the ways in which the virus can gain drug resistance. And we found that mutations in nirmatrelvir can also cause cross-resistance to other clinical compounds, but there doesn't seem to be like a single like super universally resistant version of the protease. And so with that, I'd like to thank a few people, all the great students and collaborators I work with. For small molecule screening, that was Debbie, Jiwon, and Chris. 3CL protease work, it was Show, Debbie, Bruce, and Jenny, along with Dr. David Ho. Always like to thank all the other members of the lab that make coming into work every day amazing and really fun, and the funding sources that make this work all possible. And with that, I'll take any questions.
Great talk. Thank you so much. Yeah, any questions from folks? It's a great chance to ask one of the leading experts about this, or just general questions about, I don't know, Dr. Chavez's path to this position, or.
Yeah, if people are interested in talking about being an MD PhD, happy to talk about that also.
I have a question. Wait, sorry, let me turn my camera on. My name is Matilda. I'm a data science major, so I'm not super familiar with all the like biology, but I studied a little bit in high school, so I think it's really cool. And I was curious how, like, other than sequencing, you actually collect all this data and quantify it.
Sequencing actually is one of our best outputs. So what's amazing about sequencing now is you can count like billions of things. And so DNA sequencing has sort of been like transformative for biology. Essentially, what everyone does now is they try to convert their assay into one that can be read out by sequencing, because it just has this incredible scale. The other ways that people do collect data is imaging data has now become quite popular. So you collect terabytes and terabytes of imaging data, and then you use computer vision to like analyze that. And then we do a lot of like classic approaches, you know, like measurements of densities of cells using turbidity or luminescence. But for us, and for honestly a lot of biology, you're going to be dealing with tons of DNA sequencing because it's just incredibly powerful.
Thank you.
Yep.
Yeah, and I'll post a couple links in the chat. So for folks that are in the PRISM program, so Tiana Rosing and I actually do a lot of work accelerating a lot of these tools that you use to process those reads. So you kind of sequence a bunch of data and then run it through something called a read mapper that basically figures out where do these come from. And then downstream, you can transform those results into all sorts of things that you're interested in. So, you know, especially in the lab meetings, feel free to ask Tayana and me any questions about some of the technical stuff we've been doing on that front. But yeah, it's an amazing time we're in terms of biomedical research.
Yeah, and read mappers that are good are really valuable because if you don't map the read right, you can actually get the wrong conclusions. So there are lots of people that have been mapping microbiome sequencing data, and they've been mapping it totally wrong, and it's giving them totally wrong conclusions. And that's crazy. Yeah.
Any other questions from folks? Feel free. No pressure, but no.
It's all good.
Okay, cool. Yeah, thank you so much again. It was great having you, and this was a super exciting talk. We really appreciate your time.
Sure, no, good. Yeah, and anyone's too shy and just wants to drop me an email with questions, happy to answer them.